Today, it's another genre which I suggest to your attention. It's a sort of an example how to do phylogeographic studies. It's quite rare direction, I mean, the quite rare field in our institute. But it's really very interesting and growing field which involves both molecular biology, phylogenetics, ecology, geography, and all that. I think Jimena will show this sort of an example. And I asked her to explain us some basic ideas of phylogeography. So I have to introduce Jimena. So the complete name, as you know, in Spain or in Argentina, they have a name of, well, 3 kilometers long. You know, Maria, is it all your names or you have more?
No, that's all.
Oh, that's all. Oh, that's, you know. All right. At least 4 names, that's all right. So Maria Jimena Fernandez Gomez, she is working with Argentinian Museum of Natural Science, Bernardino Rivadavia. All right. And she is here on the grant of Russian Foundation of Basic Research. Yes. This year, Russian Foundation of Basic Research organized this special competition for the young foreign scientists doing research in Russian Federation under the leadership of, candidate and doctor of science. So there are 2 persons on this grant here, Jimena and one Japanese girl in laboratory of, I think, So anyway, I have a pleasure and honor to introduce you, Maria Jimena Fernandez Gomez, and the story of Tuco-tuco. All right, Jimena?
All right, thank you for coming. This is part of my thesis dissertation.
In my
all right. I work mainly with a group of rodents from the suborder Istricomorpha. Part of this order is also the guinea pig and the chinchilla. It's the only genus in this family, Denomidae. It's one of the most species groups of rodents. It's probably 40% of the subterranean rodents. This group particularly live in the all the south of South America, and the group I particularly going to talk about lives in Argentina. It's a Perensi group, belong to one of the major lineage, the Torquato lineage. It can consist of 3 named species, Tenomys roishi, Tenomys perensi, and Tenomys dorini, and a group of forms whose taxonomic status hasn't been yet completely resolved. This group is a subterranean rodent. It has a wide range of karyotypes forms, from 2N10 to 2N70. It's mostly solitary, and there's only one exception of social species. They live in small teams, mainly in a patchy configuration. They have low agility, they move very little, they're very territorial, and they live in well-drained sandy soils and very aerated. The group I work live in the northeast of Argentina. That would be around here. The distribution is surrounded by 3 main rivers, the Upper Paraná River, the Middle Paraná River, and the Uruguay River. This is the province of Corrientes. Most of the territory is occupied for a huge westland that next to Brazilian Pantanal, there are 2 of the most biggest wetlands in South America. The western is a complex of lagoons and marshlands, and is mostly of the time, 60% of it is underwater. There's a huge amount of water, and between these marshlands there's sandy hills where this species lives. For my main work, I try to find how many species actually are in this distribution and how many populations, and a little bit about the demographic history and demographic connection between these populations. For that, we sample 26 localities in 2 time periods, from 1993 to 2003 and from 2007 and 2010. We went to that places, to the original places twice, except for one place that was flooded to make a recreational center. We managed to find another sample locality nearby, but it was 1 kilometer apart, so we treated it like a separate location. I got a total of 344 individuals. I used 2 different markers, one of fast evolutionary rate, such as microsatellites. I used 12 microsatellites, and a slow mutational rate marker, which is a fragment of the mitochondrial contour region of 411 base pairs. This is the location of the named species. As you can see, the Nomis roishi is in the northwest of the, well, I can do this. In the northwest of the distribution, with a karyotype of 4876. Tenomys perensi has 2 different nucleus divided by one of the main rivers of the area, with the karyotype of 50 and 80. And Tenomys darwini is also divided into different nucleus, one in the south of the distribution, one at the north of the distribution, with the karyotype of 2N, 70 and 80, and the rest are not quite karyotype just yet. The first thing I did was to perform a phylogenetic reconstruction to see how, if I can differentiate these species in base of genetics. They already have it done by morphology and karyotype. As you can see, there was no robust reconstruction on phylogenetic. What you can see this is this is similar to this. It means that all the branches of the tree crashes in one common ancestor. There is no differential in between the named species. Genetically speaking, it's only one species. But what I could see through an haplotype network, which is this, there are 2 groups of haplotypes that seem to be more connected to each other than to the other, and gives a differentiation that goes northeast and south, northwest and southeast, above and beyond one of the main rivers of the distribution. One of the possible explanations I found for this was connected to the geological history of the area. One of the main rivers, the Paraná rivers, at first around 7 million years, was running more to the north and east of its current location. And then it started to went down to this point, where it changed its course from east to west and then to the north. What I think it happened is during around 100 years, the species start to colonize this area, following the movement of the river from south to the north. That means going from this to up to here. And that's why I found this pattern from south to north. The next thing was try to delimit the numbers of species and populations. For that, I used an Bayesian approximation through assignment. These methods, what it does is split the sample individuals into a number of populations that the user asks the program that minimize the Hardy-Weinberg deviation and the linkage equilibrium. Then you have a probability for each possible number of populations, and then you search for the most probable number of populations, and that is what we found. Mainly what we found is a hierarchical structure where you have in these colors 12 populations that you can see colored the sample location, and lineages that seem to englobe these populations. These were the two most probable number of genetic groups that the program found. What it does is assign not only a probability to these groups, it's also assigned a probability of pertinence of each individual. And so I could assign these individuals to the different groups. Then what we are trying to do is to see how well connected these populations were or are. So I estimate contemporary and historical gene flow through the use of the two markers. Mitochondrial markers is a slow substitutional rate marker, so it gives a more historical gene flow. Microsatellite loci are a more fast evolving marker, so it gives a more recent gene flow. And also I use two different methods. The first method is you use the genealogies of these contour region and microsatellites, and it's linked the time of the coalescence event, which means when two sequences coalesce or crash into a common ancestor, and time this link this timing to different population parameters, such as the size of the population or immigration rate. And the Bayesian approach is similar to the assignment test, except that you already know which are the populations, and the only thing that the program does is to put a probability of pertinence to each individual through the analysis of the allele frequencies. It takes one individual and checks how similar it is to the different groups of genetic groups. And if the individual is similar to the one it was sampled, then it means there is a no migrant. If the individual is different or it looks similar to other genetic groups, it means that it's a migrant. So I have 3 points in time. A more historical for mitochondrial DNA, a more, that means like 1,000 generations ago, then a more new point in time, kind of 100 generations ago for microsatellites through coalescence, and the most recent, let's say 5 to 3 generations ago for the Bayesian method. As you can see, the historical gene flow shows several significant migration events between, in black, all the populations, and between the lineages with the blue arrows. Then, for the point in the middle, I only find one migration event between two of the major lineages. No migration event between the populations or the other lineages. And for last, for the Bayesian method, the most recent method, shows absolutely no migration event between our localities or lineages. This means what I found is that there are natural causes to this, and there are human perturbances that could explain this diminished in the migration events. Mainly, what we are right now is what geologists call a wet period, which means there's a lot of water in the system, and there's the Ibera wetland is mostly of the time covered by water, which difficult the movement of these kind of individuals. Therefore, the first diminished from the first to the second point in time are probably through natural causes. The second diminished was probably to human perturbances. There's been a lot of agricultural changes and forestry in the area, and there's a lot of road being constructed there. So probably there's been a lot of new obstacles for these individuals to move around the distribution. So the next step was to try to evaluate the effect of different environmental factors on the genetic structure of populations. For that, I initially used 9 environmental factors. 3 of them were obtained through the analysis of satellite imagery, which means I download the Landsat images, I position all the individuals in the image, then the images are formed for pixels, which each pixels have a defined characteristic. So I told the program which characteristics are habitat, which characteristics are water, or what characteristics are roads. And then I asked the program to interpolate this characteristic to the whole image. Then I took mean value of the sample location and an area that takes this value in the middle, and around it like a 1.5 area diameter. And from this area I obtain the amount of optimal habitat there is, the vegetation index, that means how much in vegetation cover there is in the area, and the mean elevation. For another several institutions and projects, I obtained the risk of flooding from the area, the bulk density, which is gives an idea of how compact the soil is. In the case of these animals, we need very loose soils, and the annual average precipitation. And I use 3 different measures of distance, longitude, latitude, and distance is not actually a measure of, it's more like a measure of connectivity, because it was made through the average distance between one focal locality to the rest of the other populations. And it gives an idea of how connected one focal locality is to the rest of the populations. The bigger the number, the less connected the population is to the rest. So, because many of these factors are probably correlated, I use a regressional analysis to eliminate redundant information. So I finally use for the analysis 6 factors: the elevation, the bulk density, the vegetation index, the habitat, the precipitation, and distance. I use this program, it's called JEST, that the first thing the program does is to analyze the genetic differentiation between the populations and the overall number of populations through the FSTs, and then links these differentiations to the different environmental factors. To do this, it generates models that combine these different factors from the first model, which means no factor, which means no, none of the factors that I use actually explain the genetic differentiation that is found in these populations, to the systematic use of the different factors, to the whole combination of all the factors. In my case, I use 6 factors, which means that I have 64 models. There's an inconvenience in this, is that the whole distribution of probabilities given for the different factors makes that the most probable factor that have the majority of the probabilities is reduced by the fact that the other factors have some level of probability. As a whole, the probabilities reduce the probability of the best factor. So, for this reason, I made this analysis in 2 steps. The first step, I used the 6 factors in 2 clustering levels, which means I use I did the analysis per sample locality and per the 12 populations I already have assigned. And then I used what the program called sum of probabilities, which means it sum all the probabilities that of the models that incorporate one particular factor. For example, in this case, for PG1, which is one of the factors, incorporates this probability and this probability and this probability, and is summed all together. So from this sum of posterior probabilities, I choose the 4 factors that have the most probability, and then rerun the analysis another time. So the results were, for the step 1, for 6 factors, the models that better explain the genetic differentiation between these populations were the model that include distance and elevation. And for population clustering level, the best model was the one that include distance, habitat, and elevation. Then I made the sum of posterior probabilities to choose the 4 factors with the most probability. In the case of localities, I choose elevation, bulk density, habitat, and distance. And in the case of population, I choose elevation, vegetation index, habitat, and distance. Then I rerun the analysis with only 4 factors and with the 2 clustering levels. And the results were actually the same. For localities, the best model include distance and elevation. And for populations, the model include distance, habitat, and elevation. These regression coefficients are tell us which one of the factors that are included in the model has the most influence in the genetic differentiation. As you can see, in both cases, distance is the factor that had the most influence in the localities. In this case, for this species, not only the distance is important, it's also important the heterogeneity of the landscape that they are living. Not only these animals have very low mobility, so between the low mobility and the large bodies of water and the large distance is obviously that the populations are not well connected. In the case of elevation, that is also present in both localities, in both models, it reflects the physical and energetic difficulties that life underground represents. Low soils means are more connected to water bodies, and the soils are more wet, which means it's more difficult to dig, and in the levels of oxygen are lower. So for these individuals, it's most important that the soils were well drained and well aerated. In the case of populations, habitat, which gives an idea of how fragmented the area is also relevant at this larger spatial scale, which means gives an idea in which fragmentation is an important obstacle for the differentiation of these populations. So, as a summary, I found that the name species have no genetic differentiation between each other. What I could include in these several lineages were somehow localities that have different karyotypes names, which means that karyotypes are not a barrier for the formation or for the mating of these individuals, and that and I found that many of the nucleus of the species are together with, for example, this is a named species and this is a named species, but they all come together. So I probably think that the morphology and the karyotypes need some review because genetically speaking, they are probably the same. Then what you have is that there's been a diminishing of the connectivity between the populations that can be seen through the reduced migration events in the 3 time periods. And that connectivity, which is how well the populations can interact with each other, seems to be the main factor that is affecting the genetic differentiation in this case. Thank you.
Так, вы сейчас сразу говорили, меня хотели видеть, чтобы я вам делал короткий саммари. А? Про что все было? Не хотите? Тогда задавайте вопрос.
Thank you very much for talk. But first I would like to understand where is underground life? You demonstrated some genetical process in the population of some species, but it may be everywhere, above ground, underground, in animal facility even. But where is specificity of their underground life?
I'm sorry.
Subterranean life, fossorial.
Yes.
Is what specific effect related with fossorial life?
How its fossorial life is connected to your finding. So it can be, well, any animal or, okay. Do you understand?
No, I don't understand the question.
Okay, just before, I would like to get more teaching information for me, because I don't know, I never see tuco-tuco. and I did not study this species, but anyway I experienced it in studying of underground animals. And maybe you know some names related with such study, is like Hinekburda from West Germany, or
no, I don't know much about physiology.
And I know some Argentinian professor Schleich, or similar name, I don't know exactly. Well, but is it, what reproductive system in this species?
I think it's polygeny.
Polygeny. And not like or naked mole rat or Acomis fensele or similar. It's polygeny. And they are living solitary or in
They're
group.
solitary. Mostly of them. There's only one species that is social.
And does it chisel-tooth diggers, how they produce their toenail? By paw or by tooth?
I think by tooth.
By tooth, easily. Okay. And then I would like to understand the change of environment. What population responds to these changes? Increase fragmentation, increase the problem of connection of one individuals to other, or because if you talk about evolution underground, I should understand what factors were important for such evolution, because underground it's a very specific style of life, and first of all try to get food. And apropos, what about food?
Well, they are fossorial, but they move above ground for feeding.
They are feeding above ground.
Yes.
Is it a grass eater?
It's grass and root eater, both.
Both. Grass and root. Okay, and so underground is very serious. Food, first of all, because most specialized species for underground life, they spend most time underground, and only for the few seconds we can see only no spot above the ground. And so they are using digs to produce a very long toenails and a special job, and also special demands to working system and for gas composition like CO2 or oxygen, etc. It's quite a lot of questions related for adaptation to live underground. And I would like to understand how it's connected with general observation of molecular chromosomic evolution, because I did not see any relationship again.
Well, it's probably connected through elevation, and which means that they specifically live in hills where they have sandy soils that are loose and they are well drained, and they have a good concentration of oxygen. You can find them in lower or in lower soils, lower grounds, or more clay grounds. It's like a complete line that they do not cross.
So that means that the habitats are, well, very patchy.
Yeah.
Is it?
Yes.
Yeah. So, but that's more or less the rule, that the underground animals, these mammals, they have very a lot of species.
Yeah.
Like what? Never, what?
Well, but it's several solitary species,
Yeah.
like soccer, at least, in our area, and many colonial species, and at least eusocial species, like naked mole rat, like Fuconis ancelle, and etc. Many things.
No, but here it's mainly well fragmented habitats and very low mobility, very low migration.
Well, but it's very common for many underground species.
Yeah.
And I would like to say, in my lab, We treated mole wall in those time, like in late October, and implanted special devices to measure body temperature, and returned 5 or 6 such animals in the next spring, because they stay on the same place where we were released.
So they just, like in animal house, you just put them and they stay there.
Well, but I would like to say, it may be interesting for our institute, I'm boss of a big animal facility and I'm studying mole wall, but I have no mole wall in our facility. When I did, we drove over the ischium and trap such amount which we need. We don't spend money for animals which are living around us.
More or less, they do more or less like this. They don't breed them, they don't keep them in animal health, they just go and they go along the road and they see where these tuqueiros are. Okay, any other questions?
Thank you for your talk once again. You have mentioned that the current systematics of this taxa needs a review.
Yes.
But at the same time, you say that the chromosome number between the populations are different. But at the same way, at the same time, they seem to represent not the different species, but different lineages of one bigger, maybe super species. So how do you think all your investigations, may they have any, I don't know, any consequences in the taxonomic way? I mean the species limits.
I think the taxonomy probably needs a good review because it has been made for a long time and there's new techniques that can tell us if they're actually different or it's just plasticity phenotype, right? And what I see through microsat is that they put together sampling sites that have different genotypes, karyotypes, which means that rearrangements in these karyotypes have no value as a barrier between groups. So these lineages or these big groups that I find right now are isolated between each other. They contain several sampling sites that have different karyotype numbers. If this isolation continues, they probably became new species. If somehow there are new connection between these lineages, they will probably still be like a big metapopulation where all the small groups are connected to each other, and when one group disappears, the others
they remain potentially compatible.
Exactly.
That's what we can say.
Yes, so far.
So that's another story. That's very interesting story. You notice what the fantastic differences between the karyotypes. So it's something like 40 and 80,
yes.
The chromosome number, the diploid number. It doesn't affect, it doesn't produce isolation. And that is the question why. Why is this substantial chromosomal differences doesn't affect gene flow at all? Well, you may say they are already isolated. There is no gene flow without chromosome, but no. In some places you find, we found last year a lot of chromosomal heterozygotes. Like, they have, we look at the meiosis and they produce a chain of 4. And it looked like they have absolutely normal meiosis in this complex heterozygote. And that's the point why humanity is here. So we shall look at how they manage to have a normal fertility with this very complex chromosomal heterozygotes. All right.
And also, many thanks for your last sentence, because it's very pleasure for me.
What?
I all time suspect that such a chromosomal business is rather waste exercise, and I understand it doesn't mean anything in species relationship, but anyway, I respect.
I will not sign for that.
Okay,
No.
so I would like to ask, how do you trap these animals? What methodology?
We use vectors, you know, with we condition them with
and protect for
just to get it alive, of course.
And I did it. I
Yeah.
know how to do it.
Okay.
The idea is you just open the heel,
Yeah,
and the animal don't like it at all. And you put the capcan there.
yeah.
The animal don't like it at all and come immediately to close. It doesn't look at the question.
Yeah.
So he come immediately to close the door. And you see when you don't go, you just sit near the torqueira and smoke. When you see it's there, you take it off. So it doesn't suffer. Well, 10 minutes, 5 minutes, 1 minute, 2 minutes. That's it, not more.
Sometime half hour maybe.
No, you sit for half an hour. You finish all your business.
Well, but it's Yekaterinburg methodology. I know this. They are using such method for trapping for fly trappers. They are using their hand and waiting for animals and touch by hand.
Really?
Yeah. Yeah. Well, but at the same time, I would like to say,
East German said, учиться у Советского Союза, учиться побеждать.
yeah, and there is several other live traps for such kind of species. For example, we are using very simple live traps, and we can trap approximately 20 individuals for the 4 hours. It's quite fast and very simple construction based on the wire and some special.
It's like кротоловки вот эти, да?
Yeah. Well, but other one, I study and I know this method in Minnesota. They are using for trap of gophers.
Yeah.
It's also a very interesting system. They are using long panel with rope in the distant side of this panel, and gopher, when he go inside, they found something in vertical position and try to check does it eat or not, and destroy this rope and close it by special system. Well, because it's
But they are root eaters.
yeah, root eaters, yeah. Well, and also I'm interested in what about is any MRI images of nasal area of this species, and does it possible to get it in your university?
What's an MRI?
MRI,
What's
magnetic resonance imaging.
No, I doubt it.
It's because now we are very interested in the nasal aerodynamics in underground species, and try to combine the specificity of airflow in the nasal area in underground species, and mice, and hamster, and human, and we discovered very interesting thing. It's absolutely a new idea. We found the very specific pathway for the air and protection olfactory epithelium in the nasal area. And it protects brain from nanoparticle which can move from nasal. And we are interested to increase the number of species, and if it's possible to do in any way nearby your university, we will your partner in study this, because we have very good expert in aerodynamics in this area.
And they live in sandy soil. They should be, they should protect it very well. You know, this nose. Yeah.
Well, but they should protect nose and lung from submicronic and nanoparticles, because it's serious problem, and we are studying this problem in our species. And interesting to study some other one, and especially interesting because, as far as I understand, they are using both underground life and above ground for eating.
Yes.
This intermediate is maybe interesting.
Okay, any more questions? No. Thank you very much, Jimena. Now.